Hey. So today I want to talk about just a few principles of photochemistry to set the stage for talking both about prebiotic photochemistry and present photosynthesis, what makes photopigments and features of existing photopigments, then I'll try to summarize.
Yes, all right. So yeah, presentation from Dan Kenneth from University of Liverpool. Take it away.
Yeah, thank you very much. Very nice to be here. Thank you very much for the late notice invitation to fill in. My talk will kind of follow on quite nicely from Jonathan's.
Great. So I want to introduce today a framework for thinking about ecology at all different scales that I think is useful not only for life on Earth, but also thinking about life across the universe. So as many people know, recently, it's not advancing.
In front of you. Yeah, right there.
So as many people know, these sorts of relationships relating the logarithmic size of an organism to things like its overall metabolic rate or biomass production rate have been pointed at as things that might represent laws of life across the universe, right? And it's sort of amazing in this plot that we're going from unicellular photosynthetic organisms all the way up to multicellular photosynthetic organisms, but also heterotrophs. And what's nice is that these sorts of relationships come often from the optimization of a small number of physical constraints operating on an organism's architecture. So in this case, you can imagine taking the architecture of a plant, a vascular plant broadly speaking, and optimizing it under hydraulics, mechanical stability, and the need to fill space, both to feed all of the leaves and also to fill space appropriately and take up sunlight from the atmosphere. And once one does this sort of optimization, you find that you predict a whole family of these scaling relationships. And so what this tells us is that things like the total volume flow through a tree as a function of stem diameter come from optimizing this architecture under a small number of constraints. So there are very few assumptions here, and this is the type of thing that makes us think we can trust these relationships sort of anywhere in the universe, assuming that you get to complex life like this. But it doesn't just stop here. These aren't just neat relationships or neat laws or neat regularities. They're things that we can start to build real ecology and real predictions from. So here's one really nice example where you simply take one of these architectures, you situate it in an environment where, for example, all of the geometries of the canopy tell you the total energy budget, which you can then close in terms of evaporative loss of water. And then the whole architecture of the root system tells you how you uptake precipitation from that same environment. All of these things are simple equations of the overall size of an organism. And then you can create things like these survival phase diagrams for plants. So here, for example, is putting everything in volume flow rate. The basal needs of the single cells to survive give you an overall flow rate through the plant for just simply not to die. And then there's this total amount of water that you can get from the root system that's set by individual environments of different precipitation. And then here's the actual evaporation of the canopy. So anywhere in this green space, a plant is happy to survive. This is the ultimate limit to plant height in a particular environment. And then the black curve tells you what's actually happening for a canopy. And one can then start to do things like simply solve for where this curve intersects one of these two curves, predicting a maximum possible height. And if you take known variations, say at the continental scale of different climatic features that matter for this budget, you can very simply, with a small number of equations, predict the variation in maximum tree height across a continent. So these are observed maximum tree heights from ground measurements, and these are the predicted maximum tree heights we get from this very simple framework. These geometries are not only useful for this type of broad-scale ecological prediction, but you can also predict all the local plant traits as a function of these same variations. So it's very simple to derive, for example, from this architecture, how things like canopy albedo should change with tree height. And then it's also possible to predict how the individual plant traits like the stomatal density or leaf size should vary with features like temperature. And so what this gives us is a really simple way to go from broad-scale continental variation to a whole suite of plant traits and remote sensing features that we might be interested in. There's also really nice work showing that you can also take these sorts of scaling relationships and build demographic models. So if we think about how all of these different features change with plant size, and we think about a distribution of plant sizes, one can then solve for the steady state distribution and broadly predict that you get these power law distributions in tree size within a single forest. These, of course, have been criticized for not appropriately capturing the tail of the distributions in lots of really real observed forests, where you had this nice power law behavior at small size, but then this sort of sharp cutoff for large size. And we showed recently that that is simply a consequence of environmental variability. So if you take one of these demographic models and you simply add an environmental variability term, then you get a more complicated form than a power law that has this exponential cutoff. And that does a really nice job of predicting the observed distributions, and then also the temporal dynamics as forests recover from disturbances back to one of these steady state distributions. And so I'm making quite a lot of these distributions, and the reason why is because they allow us to do things like simply predict whole forest features once we know these maximum sizes in the distribution. So here's work we've done showing how biomass density, net primary productivity, scale with the maximum stem radius of a tree. So that is, if we know the maximum size, we can immediately predict biomass density and net primary productivity, and we can predict the relationship of the two with each other. And so what this really gives us is the ability to scale up to sort of any scale we're interested in. We know how plant traits scale with size. We know how distributions then scale within a forest. And we know how to scale those distributions across environments of different maximum size. And all of this really is a way to go end to end from local environmental conditions to forest structure. And the thing I really want to highlight here is that all of this is highly nonlinear. So for example, the way you scale up NPP from biomass involves this 1/2 power rather than a simple linear scaling. And so you might need to know both local environmental conditions, but also this nonlinearity to appropriately scale biomass up to any given scale. And then finally, I'll end by just saying that we've also generalized this. So trees are a very particular type of photosynthetic organism. These large vascular plants, we have many others. We had many others in the history of Earth. But what one can do is take these demographic models and situate them in these sort of more complicated scaling regimes where you have sort of features of competition, how fast things grow as a function of size, and how fast they die as a function of size. And if you scan within this parameter space, we've been able to situate all of the different sort of biological systems of interest, showing that you can go from sort of randomly packed forests to hexagonally packed things like corals or termite mounds. And all of these will have different macroscopic scaling features. They'll have different biomass densities. And so it's important to sort of think about where we think we are in the history of life, what type of organism we think we have, but this gives one a framework to at least scan over these parameters in a very general way and start to build expectations for total biomass and the like. Everything we've done here, we've also done for microbes. So this is just a teaser for that, but these same sorts of frameworks can also be applied for the whole range of microbial life. And with that, I'll just end by thanking a set of really wonderful collaborators and a bunch of really wonderful funding support. Thanks so much.
Okay, we have time for a couple of questions online. Just a reminder online, type your questions and we can read them out here if you have them. We'll scan for that. Let's start over here. Remember to introduce yourself.
Hazel Barton, University of Alabama. The question I have for you is on when you were modeling like the current height of trees versus what we actually measure, does it account for the American chestnut? So those were ginormous trees that went extinct. Do you see a pattern for those even though they no longer exist in that ecosystem?
We do. So you can actually, the places where we are off in our predictions are either very extreme environments where plants are very specialized in terms of their adaptations, notably arid environments in the Southwest. or in places where there's been huge human impact, and we actually predict bigger trees than are currently observed because of clear cutting or recent extinctions and so forth. So the Northeast, for example, should have much taller trees than it does. That's likely all human impact. Yeah, great question.
Bob Blankenship, how does your model work for something like a saguaro cactus, which has this really different architecture from a normal tree?
Yeah, so we can, so cacti, allometries are known, and you can situate them in this space. So I don't have time to go into all of it, but there's ways to think about what the different set of constraints are for something like a cactus. There's also evolutionary dependence on what types of vascular architectures are discovered at different times. Some of those are more and less optimal in different environments. We can have a longer offline conversation, but it's basically how you situate it in this space that would then immediately lead to all these downstream predictions. Yeah.
Okay, we have one more. Nancy, one.
Thanks, Chris. Really amazing work. So I'm wondering, when you take a forest ecosystem around a GD-type star like our sun and move it to a different kind of star like an M-dwarf, what does that impact on your scaling relationships, that change in flux and spectral energy distribution? How does it factor in?
Yeah, so in our model, the things that matter are basically the environmental conditions and then the physical constraints. So for example, if you change just the gravitational force of a planet, that changes all the allometries in a predictable way. And then everything else that you would change about total energy flux would really be just how you close the energy budget for leaves. But you can imagine gravitational fields that are large enough and fluxes that are low enough that you can't support these large multicellular organisms that lift off the ground because it's just too hard to pump fluid. However, you still could get things like lichens, mosses, biocrusts, all the sort of previous iterations, and that would just, again, be in a different part of that phase space of parameters.
Okay.
Thanks again.
Chris again.